Hello, my name is Raeanne Tegman, and I worked with Dr. Gabrielle Stryker in the biology department, as well as the McNair program, on my project titled Comparison of Lyme Disease and Rocky Mountain Spotted Fever in Free-Living Ticks and Ticks Found on the Southern Alligator Lizard in Washington State. So why are we interested in this? A nymph is the immature form of a tick. When a nymph is infected with Lyme disease or Rocky Mountain Spotted Fever, and then feeds on the southern alligator lizard's blood, the infection is suddenly cleared. We are unsure as to what causes this, which makes it a very interesting point of study. A couple of important sub-questions were considered when thinking about this project. First, which group of ticks will carry more disease? Those found feeding on the southern alligator lizard or those found free-living in the environment? Second, what will the most common disease be, Lyme disease or Rocky Mountain Spotted Fever, between both groups and individually? And lastly, what diseases are being cleared in the nymph by the lizard blood, and what diseases are not being cleared? You might ask how this data will be collected. Well, first, tick samples will be collected from several different locations in Washington State, such as here in Kittitas County, and also down in Klickitat County, which borders Oregon and is actually where the southern alligator lizard is found. I would then extract the DNA from each of the ticks sampled using a Zymo DNA extraction kit. Once the DNA is extracted, pathogen DNA would be amplified by performing diagnostic PCR and then confirmed by gel electrophoresis. Any samples containing pathogen DNA confirmed by gel electrophoresis will be sent to a lab in order to confirm the pathogen DNA. So, this was all my plan, but unfortunately the pandemic caused quite the disruption. It was no longer safe to travel and collect samples of ticks from different parts of the state, so I had to improvise and think about what I could do that was safe but also interesting. So Dr. Stryker and I came up with a new idea. I was going to collect ticks in Kittitas County and test them for Rickettsia rickettsii, the bacteria that causes Rocky Mountain Spotted Fever. This led to a new question being proposed. Are ticks in Kittitas County, Washington, carrying the bacterium Rickettsia rickettsii that causes Rocky Mountain Spotted Fever in individuals? So first, a little background. Rocky Mountain Spotted Fever is caused by the gram-negative intracellular bacterium Rickettsia rickettsii. It was first discovered in 1896 in the Snake River Valley of Idaho when it was nicknamed black measles due to the discoloration of the skin during the later part of the infection. When an individual becomes infected with this bacteria, most likely through a tick bite, they can become nauseous, they can start vomiting, and acquire fever and chills, as well as this spotted rash that you can see here. In total, I collected 21 ticks. Some were found while hiking the Teanaway Ridge, while others were donated from individuals in the community. There was a pretty good mix between male and female collected, but a large majority of the ticks collected were Dermacentor variabilis, also known as the American dog tick. Here are just a few of the images of the ticks used in my project. You can see that most of them are the American dog tick. The females have this distinct white scutum, also known as the dorsal shield. Only one of them is the Ixodes pacificus tick that is capable of carrying Lyme disease. We did test this tick to see if it was carrying Borrelia burgdorferi, the bacterium that causes Lyme disease, and it did come back negative. In order to test for rickettsia, I had to know exactly what I was looking for. My target gene was a 190 kilodalton antigen. I made a 25 microliter primer mix to use in PCR and amplify the DNA. This consisted of 12.5 microliters red tack, 10.5 microliters of water, 1 microliter of tick DNA that I isolated using the Zymo DNA kit, and 1 microliter of primer. I used the RR190.70P and RR190.602N primers to look for these sequences. I then ran my PCR reactions to these guidelines, which state 95 degrees Celsius for 3 minutes initially, then 40 cycles of 20 seconds at 95 degrees Celsius, 30 seconds at 48 degrees Celsius, and 30 seconds at 72 degrees Celsius, while ending the procedure with 7 minutes at 72 degrees Celsius. After completing my PCR, I ran gel electrophoresis in order to confirm if pathogen DNA was present. The gene size that was amplified in PCR was 532 base pairs. So, if pathogen DNA was present, then I would expect to see a band around this location. Here, we can see that there was a very bright band around 532 base pairs from TIC6, indicating that it was potentially positive for rickettsia. There are many extra bands on the gel, as you can see here, likely because of non-specific primer annealing at too low of a temperature. So, I decided to do more PCR and alter the annealing temp solely with TIC6 since it does look like it tested positive, in order to see if the extra bands would clear up. This is my gel showing the different annealing temperatures that I used. You can see that there are still some extra bands appearing at the lower temperatures, but the closer we get to 60 degrees Celsius, the more that they clear up. This shows that 60 degrees Celsius is a much better annealing temperature for this primer and should be used moving forward in the project. We can also see that TIC6 is definitely testing positive for rickettsia since this band is appearing multiple times at the same location. Here I have included some DNA quantitation findings that were found after isolating the bright DNA bands from the gels that I had completed earlier. The ITS-1R DNA towards the top of the chart involved a primer that was used to identify tick species. Here, we are really interested in row D because it is quantitating how much DNA was found in the gel from TIC6 at 60 degrees Celsius from the previous slide. It is showing that a large amount of rickettsia DNA was quantitated from TIC6, furthering our conclusion that TIC6 did indeed carry this bacteria. Since so much DNA was found, we were preparing the sample to be sent off to another lab for confirmation, but due to the pandemic, this was unfortunately not possible. Moving forward, I would like to continue testing for rickettsia in the remaining ticks since I was unable to finish testing them all due to the pandemic. I would like to rerun ticks 7 and 8 as well because they did show a possible band around 532 base pairs, but since the annealing temperature was a little off for that PCR and gel, I think that those samples should be rerun. And lastly, the main project of comparing Lyme disease and Rocky Mountain spotted fever on free-living ticks and ticks collected on the southern alligator lizard that I talked about at the beginning of this presentation should be continued when it is safe to do so, because it would be very interesting to confirm that the southern alligator's blood does in fact clear the nymphs of disease, which also opens up new areas to study how the lizard's blood does this, if that is in fact the case. I would like to say thank you to Dr. Stryker for being an amazing mentor and for always being compassionate and patient when working with me. She really pushed me to reach my full potential and taught me so much about working in the research lab. I am so grateful. Thank you to the McNair program for not only the funding of my project, but also the endless support that I have received during my time at Central Washington. Thank you to Kylie, Paul, and Clayton for always listening to me ramble on about my project. Also, a final shout out to the whole biology department at Central for creating a sense of wonder and curiosity in me about the world around me that I will carry with me forever. Thank you.